This is the Kratom Science Podcast. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. They published the Kratom Genome, and my guests today are two scientists who worked on it, Julia Brose and Dr. Robin Buell from the Department of Plant Biology, Michigan State University. Thanks a lot for joining me on this. We are excited to see that the Kratom Genome has been published. So I guess if you want to go ahead and introduce yourselves and just explain generally what you do, and then we can get into the paper.
All right, I'm gonna let Julia go first.
Oh, okay.
I always let Julia go first. She's the student.
Okay. So, hi, my name's Julia Brose. I am a second-year grad student in Robin Buell's lab. I'm interested in genomics and biochemistry. So, Kratom is the perfect combination between the two, and I really enjoyed working on this project.
Awesome. And Dr. Buell?
Yeah, I'm Robin Buell. I'm a faculty member here at Michigan State University. I've been doing plant genomics and genetics since 1999. I've focused on working on a variety of crop plants like rice, potato, sweet potato, but I've also worked on a whole set of medicinal plants, and some of those include Madagascar periwinkle, which is a source of an anti-cancer drug. I've also worked on a lot of mint species, and I've been working on Kratom now for the last few years in collaboration with several biochemists.
And what made you want to study Kratom specifically?
Julia, I'm always going to let you go first.
Okay, I'll always go first.
Anybody jump in?
Yeah, I think that's the easiest thing, and I can always just add commentary because I want Julia to get credit. This is her paper. Yeah.
That's okay. So I think the reason why I started working on Kratom is because of the biochemistry background. It has a lot of very interesting metabolites like Mitragyna, which is obviously what the community is really interested in right now. And this was a project that was near its completion and it needed a final push. And I was really happy that I could have the opportunity to do it. So it was good timing on all parts.
So I'll add that, so we started with this project with Kratom because we were interested in several plant species that are in this family known as the coffee family because they produce alkaloids. So our collaborators, two biochemists, are very interested in alkaloids. And so this Kratom produces a really interesting set of alkaloids. And so this is the one that was chosen to look at clearly because, it clearly has human health effects.
Mm-hmm. And so just in general, I'm just going to go from like a really basic standpoint. And so it says, you report on the draft genome sequence and annotation of Mitragyna speciosa. Could you explain just in general what, is it called mapping a genome or kind of what that is?
Okay, so a genome is essentially all of the contents in a single chromosome. So what this paper does is we took DNA and then we isolated it and then you assemble it. So you take all of these little pieces and you want to put it together, which leads to some problems because it is a complicated process. But that's essentially what you're doing is you're taking all of the genetic information and then making it into one representation of the genetic components.
So maybe for other people in your audience, think of it as the blueprints, right? So the genome is essentially the blueprint for how a kratom grows and develops, right? And makes these interesting compounds that people are interested in. So it's just the guidebook for how a cell works and how it makes these molecules.
Yeah, and I definitely want to get into, where this knowledge that you guys, discovered is going to take us, but I want to kind of go through the process. And then, okay, so we were talking about the family that it's in, and this paper is really nice. I'm going to link to it, and I think it's open access, and I don't think I got it through my school, but
it's open access.
Yeah,
it's open access.
Awesome. And it has a nice little diagram there of the Rubiaceae family.
Rubiaceae.
Rubiaceae. I keep saying Rubica as I read it, and then I looked it up, and I didn't memorize how to pronounce it. But Rubiaceae. So, and you talked about the Rubiaceae family, and it also contains cacao, which makes chocolate, wine grapes, tomatoes.
Oh, no, I think,
coffee.
And cinchona, like quinine, yeah.
Now, is Kratom obviously it's unique, but is it more unique than the other plants in this? And a lot of these are consumable things that we all. Kratom's related to the coffee plant. Its effects are sort of similar, and just kind of what they do to the body. It's kind of like something you have in the morning if you take a nice light dose of it, and I guess just if there's anything interesting that you want to talk about, just about the Rubiaceae family.
Actually, Brian, I can't believe I didn't catch this mistake. The cacao is not in the Rubiaceae, yet it's in our paper that it says it is. It produces alkaloids, though.
Oh, okay.
Yeah, so that's a mistake on our part. I can't believe I didn't catch this.
Where is that?
That's in the introduction. Yeah.
It's on the chart,
family,
too.
It's in...
Yeah.
Coffee
I think it's just... Coffee is, but just the way it's written. Yeah. Yeah. It's the way it's...
But not term aroma.
Yeah, it's not, though. Poorly written the way it's written, right? Because that sentence is supposed to be talking about really important alkaloids, right? Which are the chemicals of interest.
Yeah.
Yeah.
But coffee also makes alkaloids.
Yes, yeah.
Which is caffeine.
Yeah.
Caffeine's an alkaloid.
It's cacao, the chocolate is outside of the Rubiaceae, but coffee is within the Rubiaceae.
Yeah, and they're both producing alkaloids.
So the whole process of studying this, you have, I'm sure you're in plant biology at Michigan State. I'm sure you have a ton of greenhouses, but you have one that has kratom trees growing in it, and these are rye fat trees that you got from World Seed Supply. And you kept them at a certain temperature and a certain amount of light. And this specific tree was named for Claude Rifat, who was kind of like one of these ethnobotanist type of guys. And do you know if these are the same strain that are grown in Indonesia that most people in the West consume in kratom products?
Julia, you want to answer? Or you want me to answer this one?
You answer this because I wasn't here when we got the plants.
Yeah.
Oh, okay.
So that's an excellent question, right? Because you just order these things online and they say, I call it Riffit. I don't know if that's right.
Yeah.
Or it's called Bumblebee, or we actually ordered some other ones and they just say Indonesia or Malaysia. So to be honest, you have no idea what you're getting, right? You have absolutely no idea. It could be the exact same plant, right? So that you're going to get the exact same genetic makeup as, say, the dried leaves that people are purchasing, and it may not. But that's not a unique feature of kratom, right? Other plants, you just order them from a nursery or you buy them, and they don't always have to be completely identical to what the name is, right?
Are the trees still growing there, or are they going to be subject to future studies? Or
so I'm actually leaving Michigan State University. I'm not taking it with me. The one we sequenced actually died.
Oh, okay.
It died in our greenhouse. But we have a second plant we ordered at the exact same time from the exact same nursery, and that's going to stay here with my collaborator. So.
Cool. And I was talking to Dr. Sharma from University of Florida, and he studied the alkaloids, so it's a little different. And the age of the tree seems to affect the alkaloids, and even like where they grow native, they're different in Thailand, Malaysia, Philippines. How old was the tree, and does that have any effect on what comes out when you look at the genetic?
Julia, you answer.
So I'm not sure how old the tree was when we collected the tissue.
Probably 1 year old from the nursery. So maybe it was 2 years old by the time we isolated the DNA.
Okay.
But that shouldn't affect the genome at all. Like whether you get it from an old plant or a young plant, you're going to have the same genome because this is, like Robin said earlier, the blueprint to the plant.
Okay, so the one part says you had immature leaves, mature leaves, leaf bracts, roots, stems. So why is it important to have all these materials and to look at instead of just one part of the plant?
So all of these different tissue types were used a little bit differently than the whole genome sequencing. This was for RNA sequencing, which is for expressed genes only. And this was used to create the annotation. So to decide which genes are active or what do you even call a gene in this genome, that's the whole point of annotation. And that's what we did with those tissues. And then we also used that to map back onto the genome to see how much of these proteins are actually found in the genome. And
there was a section called genome assembly and scaffold filtering. So what is a scaffold even in genomics or genetics or?
So a scaffold is a grouping of sequencing that when you assemble it, you're not really sure where everything goes because you have all of these different parts. But with this 10x library, you're able to link different regions together. And so those would be called contigs. And then you can further link them together to form these scaffolds, which are not whole chromosome length sequencing. They're smaller than that. So that's what a scaffold is. Just a larger section of DNA.
And then I'm just kind of going by section. And then you do like a quality assessment. Is that just kind of to check everything, make sure everything checks out?
Yeah, it's to make sure that what we're assembling is actually reasonable and the lengths make sense and that you don't have anything erroneous in the assembly. So yeah, it's just a good way to check.
Cool. And then like gene annotation, that's basically what is that part?
So that part is when you take all of your different like RNA-seq data, which provides you information on which genes are expressed, and then you use different models to model which genes are actually there, if that makes sense. So you're taking this data and then which proteins are expressed, and then you want to know which of these are actually real, and that's how you annotate it. So you have a collection of RNA-seq.
Cool.
So if you wanted to, I always like to use this example, and people can understand it. So imagine if you took all the words from Dr. Seuss's The Cat in the Hat, and you put all the words together with no spaces and no capitalization and no punctuation, right? And then, so you wouldn't be able to read what the book was, right? Because you have just 26 letters in a string.
Yeah.
And so what we do when we do annotation is we want to understand what that DNA sequence is telling the plant. So those are in little packets of information called genes. And so think of the annotation process as taking all the letters for the entire Cat in the Hat that have been squished together and then making sentences out of that by adding a capital letter, a space in between the words, and then a punctuation.
Cool. That's a great way to explain it. I like that. That was awesome. Yeah, because I'm like, I kind of know what this stuff is, but I'm like, kindergarten when it comes to this stuff.
No, I think you're doing great. I'm impressed. Yeah.
it's really interesting to me. It's just one of those. It's one of those papers. Like sometimes I can understand. Some, I'm just all learning this stuff, and it kind of works out for the podcast because people are learning with me. Then we get into monoterpene indole alkaloid biosynthetic pathway. An alkaloid biosynthetic pathway. What is that?
So these are all of the different steps it takes to make this final product. So like Mitragyna isn't just formed through one step. It's part of a pathway, a larger part of it. So this like monoterpene indole alkaloid biosynthetic pathway has many, many genes, which then code enzymes, which then have a special function to create this novel product.
Mm-hmm. I definitely understand that. I'm starting to understand about the metabolism and stuff. Mm-hmm. And I guess that information in general, will show you how the actual substance works when somebody takes it, so maybe, like a practical application would be if you understand that, then you might know if somebody has a certain condition, you might not be able to take this medicine is based on how it metabolizes in the liver. Is that accurate?
I would say it is, but it's in reverse, right? Mm-hmm. So you're used to having the final compound, and then you figure out what it does in the human. Think of it as how did that compound get started, right? And we use similar techniques to figure out how the compound was actually synthesized. So you're interested in what happens to it once it gets into a human. We're interested in how did the plant actually make it from scratch, right? Because the plant had to make it from scratch.
Yeah. And alkaloids in plants in general, we think of them as like, oh, caffeine makes me feel good and kratom tea makes me feel good. But they actually have a function in the plants, don't they?
Yes.
I know some of them. I know in tobacco, nicotine acts as a pesticide, I think, against mites or something like that. Do we know what the kratom alkaloids act as, or even coffee, or?
I don't think it's known.
I haven't read anything about it.
Yeah.
I've heard.
But we know that most of these like secondary metabolites do have a defense. Yeah. Some sort of defense compound, suggestion.
Yeah. So another one that it's in the same class of monoterpene indole alkaloid is camptothecin. And that's also another anti-cancer drug, and it's made by this plant called Camptotheca. And that compound actually inhibits this enzyme called DNA topoisomerase. So your DNA is all wound up into a helix, and you have to unwind it to do things with your own DNA. And so that compound is produced. And so if insects or animals eat Camptotheca, right, it actually is quite deleterious to them. But the plant actually has a mutation in its own DNA topoisomerase, so it's immune to it. So they're most likely functioning in defense, but we just don't know what exact insect or animal or something.
Yeah, could it be a fungicide too? I think I had Dr. Przelik who's done some kratom studies on, and he said we think it might be a fungicide, but we don't know. He also said we don't know.
Yeah, I don't think people really know for sure. But,
yeah. And kratom's kind of a new thing that's just being studied. Is that, I mean, I know there are studies going back, when they first discovered it in the East. Is it like a relatively new plant that's being studied?
So I think on our end, it's relatively new. If you look at most of the studies that we've cited in this paper, they're all within the past, 20 years? Mm-hmm. So I think the funding for it is what's changing. And, funding drives research. So if there's more funding, there'd be more research.
Yeah. But I think there's quite a long history of its use, right, as a herbal remedy.
Yeah.
So there's definitely been studies on it, but not necessarily in modern scientific labs.
Or in the Western world.
Yeah.
Yeah. I think that's important to note, right? We're looking at it because it's already been established or in a culture. So the question is can we use, as you said, modern methods and then definitively identify what the effects of it is? Yeah.
I'm going to read from the study again. It demonstrated that M. speciosa is a tetraploid and has conserved loci involved in specialized metabolisms that can be harnessed to identify loci involved in monoterpene indole alkaloid biosynthesis. Yes, I got through that. But terploid, does it mean it has 4 sets of chromosomes?
Yes. So tetraploid is you have 4 sets of chromosomes. Yeah. So for reference, we're diploid, and kratom has double what we have.
Of humans.
Yeah.
Yeah, we're diploids, they're tetraploids.
And what does conserved loci mean?
So conserved loci means that there are these, you have genes that are conserved between the duplication and with other species is what we're referring to in this monoterpenoid alkaloid statement, I believe.
I'll just add a commentary that, in the biosynthetic pathway, the first steps will be quite conserved with other plants because I believe every land plant produces monoterpenes. But then those, once you make what we would call the scaffold, the core part of the molecule, that's going to be conserved. But then the rest is going to be unique to kratom to some extent. Yeah.
And it contains 72 clusters of specialized metabolism genes, which will aid in improving our understanding of the evolution of plant specialized metabolic pathways. And that means with plants in general, right? That doesn't just mean with the kratom plant.
So this analysis was done using this Planty Smash program, which predicts metabolic clusters. So it's not necessarily saying that these are the active clusters possible. You would obviously need some experimental evidence that this is what's actually going on and that the genes identified in this Planty Smash are doing those activities. So I think you have to consider that these might not actually be present. I mean, they should have alkaloids and cytochrome P450s, but it's not necessarily that these ones predicted are actually doing the activity.
Right. So let me just add, so these types of enzymes that do what we'll call the decoration, right, are the tailoring enzymes. There's hundreds of genes in kratom in all plants that have this generalized function, and you just sort of have to figure out which one is doing which step. So it's a bit of a fishing expedition to figure out exactly which one.
So in the paragraph above where there's a supplementary table S6, that contains all of these different genes that could possibly have these MIA be part of the monoterpene indole alkaloid pathway. But because when you look at these genes, some of them can be very similar, and you're not sure which one actually has the activity until you go test it.
That's cool. And I was just going to get into kind of what kind of this knowledge is going to be, how it's going to be practically applied, because it looks like, kratom's going to become a medicine. It's the popularity of it, and if the demand's going to increase, there's probably going to be a domestic agriculture for it, which it's going to have to rely, I think, on plant biology because it doesn't grow in the regions that are most of the United States. But so there's one in the specific question I had is you talked about bioactive alkaloids and heterologous hosts. So and I looked up heterologous expression and how does that work in the science?
So the idea is much like how insulin is now produced, where you put it into yeast. You can now make insulin without actually needing to isolate it from an animal. And the idea with this is that you could take the genes involved in the pathway pathway and then put it into a vector that's not even a plant, and then produce these without needing to have these kratom plants or to grow them agriculturally, because you could put them into something else.
Yeah, so you could actually just grow the alkaloids, like.
Well, you could produce them in a plasmid vector. So you could take each individual enzyme of the pathway, or you could feed it certain metabolic products along the way. And then you could have the end product created by not a plant, but through this vector. Mm-hmm. If that makes sense.
Yeah, that totally makes sense. That is cool. So understanding the biosynthesis of the bioactive alkaloids, and it talks about enabling more precise pharmacological studies on the positive or negative outcomes of this medicinal plant, and eventually inform breeding and production strategies for this species. So that would be like the other path of, learning more about how the plant grows.
Mm-hmm. So you could target certain genes or breed. Like you discover, this variety has like a higher Mitragyna like content, so maybe you select on those to breed further. I think it provides you with that.
So Brian, just back to your other question about what you would do with this genome, and your also question is the riffet we're growing the same riffet that's being produced? So now that we have the genome, you can do this essentially genotyping. It's very similar to 23andMe, right? We could see whether or not all the riffets, like let's say I bought a riffet from like 10 different nurseries online, are they all the same, right? So once you have the genome, that'll help you figure out whether or not everything's the same or not. And then you can also figure out if they're different. Say, like we also have this accession called Bumblebee, right? If they're different and they're different genetically, are they different in their alkaloid profiles, right? Mm-hmm. And this breeding aspect, so I'm not sure if you're familiar with it, but opium poppy has been bred to have different opioid profiles. So some poppy cultivars would produce high amounts of morphine, some may produce high amounts of codeine, some may produce this noscopamine. So you can breed and select for unique profiles of your target compound.
Yeah, and I'm sure like our audience is familiar with, there's one plant you can grow in pretty much every state that produces CBD, and the other hemp plant, produces marijuana. So that's kind of, I guess that's kind of the same. Is that similar?
Yeah.
Was there anything else on this paper specifically you want to talk about, or?
No, I think it's a good resource for the community.
Definitely. And I'm just looking at your lab and just plant science in general. Like you're doing work on potatoes and all kinds of stuff, beans and you're talking about like drought tolerance and corn and stuff like that. I was a farmer for 7 years, so you kind of have to be a bit of a plant scientist when you're a farmer, because your livelihood depends on it. But can you just talk about like that a little bit? Like, what does plant science do? Does it help increase yields or grow more drought tolerant food crops? Or, I mean, I'm sure it does all that.
So you want an answer in terms of like my lab specifically, what I've been doing?
Yeah, sure.
Or just, yeah. So I'd say, and you're interested in this bean work, so maybe I'll just talk just about that bean work. So common bean, right, which is like pinto bean, kidney bean, black beans, navy beans. They're very susceptible to high nighttime temperature. And so with climate change, that'll have a big impact, especially in developing countries, on their bean yields. And, that's their protein source, right? They don't eat meat, they eat beans. But, because of evolution and adaptation, there's a sister species, a very close relative of common bean that evolved in the Sonoran Desert, which is obviously hot and dry. And so we studied its relative. It's called tepary bean, because it can grow in really hot conditions. And we wanted to ask what's in its genome, and can that give us clues as to how it can deal with hot and dry conditions? Because if we can understand those, then maybe we can focus on bringing those traits into common bean and making common bean heat and drought tolerant.
Wow, that's pretty cool. Like it's we started a small farm with my ex-wife at the time when the kind of organic farming was big, and a lot of people kept asking me if the seeds were GMO, were they GMO, and I'm like, I don't know what that means. And then I looked into it. I'm like, it's actually really cool. I'm supposed to be like, there was a kind of like against GMO thing. I mean, do you have any thoughts about that? Like, I know there's a section on gene editing, and there's kind of, I think there's a lot of misinformation about what's actually occurring there.
I'll let Julia comment as a young person on GMOs.
That's fine. Well, I think it comes down to what you consider as a genetic modification, because we've had years and years of breeding and domestication, and that's significantly altering the genetics of an organism. Mm-hmm. So if you consider that genetic modification, like everybody's doing it, that's how we survive.
Yeah.
But introducing different genes from like foreign, you would call them foreign DNA, into a plant for some novel effect or pesticide resistance. At the end of the day, I think that if you're going to feed people, you need to feed people. And that's what it comes down to. And science allows us to feed more people and to adapt to what's going to happen in the future with our climate. So you
absolutely,
there's a point where you need these things, and science allows us to do it, so we should invest time in that and consider doing it.
That was Kratom Genomics. Thank you, Julia Brose and Dr. Robin Buell from Michigan State University Department of Plant Biology. The music is Risey. The song is Memories of Thailand. The Kratom Science Podcast is written and produced by me, Brian Gallagher, for KratomScience.com. Please like, subscribe, review, and take care.